Yeah, good morning, everyone. Again, it's me. I'd like to thank the Q Cancer team once again for this great privilege to be here. In this section, I'd like to share, our recent research on the cytokine, which is very important for our immune system, interleukin-21. And I will start by introducing a bit about the background story of this research. So we are a T-cell immunology group, and why is T-cell so important? Because the major function of T-cells is to directly kill the pathogens and tumors, and this is mediated by a specific subtype of T-cells called killer or cytotoxic T-cells, and they express CD8, so we also call them CD8 T-cells. However, CD8 T cells in cancers are very prone to a dysfunctional state, terms exhaustion, as they have been persistently exposed to a very big amount of tumor antigens, and they also received the immune checkpoint inhibitory signal from signaling such as PD-1. So the exhausted T cells will gradually lose the function and cannot effectively control the tumor growth. But we all know that in the past decades, a major breakthrough in cancer therapy is that we found if we can block those inhibitory signaling, such as PD-1 and CTLA-4, and then we can effectively revitalize T cell function from exhaustion. And this strategy is called immune checkpoint blockade. I'm sure we're gonna listen, we're going to hear more about these regulatory pathways in the talks today. And they really have prolonged the survival rates in many cancer patients. But as we can saw from many researchers, in most of cancer types, the efficacy of immune checkpoint blockade therapies remain to be improved. And this issue is highlighted by what we saw in lung cancer, so which is the most common cause of cancer death in Australia. So in lung cancer, in lung adenocarcinoma especially, which is the major form of the lung cancer, the overall response rate to the immune checkpoint blockade is below 20%. So the major question we want to address here in this Q Cancer project is to identify a new approach so that we can boost the T cell immunity, improving the cancer, the immune checkpoint blockade therapies response. So to address this question, we need a more comprehensive understanding of the anti-tumor immunity beyond just CD8 T cells. So in the tumor microenvironment, there are also CD4 expressing T helper cells, which literally provide essential help signals to CD8 T cells, and they are also very important in affecting cancer patients' overall survival rates and their response to immune checkpoint blockade therapies. So in particular, this specialized subtype of T cells called T follicular helper cells has been demonstrated to be required for the anti-tumor immunity in lung adenocarcinoma. In this recently published Cell paper, the authors identified that if we increase TFH infiltration in the tumor, is positively associated with the better survival in the patient. But TFH cells' role in anti-tumor immunity wasn't really extensively studied before, because this population is more commonly associated with the antibody response by regulating B cell function. But more evidence suggests TFH functions are important for regulating anti-tumor immunity. So my previous research in TFH cells biology is also focused on the B cell immunity side. We generated a couple of new discoveries. But what we want to emphasize today is that the way the TFH cells contribute to enhance the anti-tumor response is by secreting a very important cytokine, interleukin-21, IL-21 again, to the CD8 T cells. And as we can see here, if the CD8 T cells have defect IL-21 signaling, that will very severely impairs their killer function in the mouse lung cancer models, as we can see here. So that's why, since IL-21 has been identified, it has held significant clinical promise. And IL-21 alone or in combination with other drugs have been tested in clinical trials. However, the overall response rate is not very satisfactory again, which is not too surprising because IL-21, just like other cytokines, they tend to have very low in vivo efficacy. But if we increase the dosage, the dose-limiting cytotoxicity will come first. So how can we find a way to improve IL-21 efficacy is the central question to overcome this limitation to unleash IL-21's therapeutic potential. So in our recent work, we identified that the bioactivity of this cytokine is actually regulated by a conserved cationic residue cluster, meaning these clusters of enriched of positively charged amino acids, as we can see here. And we try to generate a mutation to change these positive charged amino acids into a neutral amino acid. And what we found is that by this kind of mutation, the function or bioactivity of interleukin-21 has been drastically increased. So that indicates that physiologically, this is an important mechanism to regulate IL-21 signaling in immune cells. But it also indicates that this CRC region could be an important target we can harness in the engineering and improve the IL-21 activity in vivo. So by harnessing this biochemical feature of interleukin-21, we developed an antibody that can specifically target the CRC region. And as we can see here, if the IL-21 doesn't have the CRC region, then their binding towards this antibody can be reduced by over 700-fold. So indicating how important the CRC region is for this antibody binding. But more importantly, functionally, if IL-21 forms complex with this antibody, then we can see its efficacy to promote human CD8 T cell activation has been drastically increased by over 10-folds, as we can see here. The activation of CD8 T cells was measured by the CD25 and gram beta positive T cells. Molecularly, that's also because when IL-21 forms the complex with the antibody, it can induce a more prolonged STAT3 signaling in the T cells. As we can see here, the EC50 of the IL-21 antibody complex to induce STAT3 in CD8 T cells reduced by over 10-folds, as we can see here. So we were then very interested about the mechanism of action of this antibody. So we actually solved the structure, crystal structure of interleukin-21 in complex with the antibody Fab fragment. So we further overlay the structure of the the antibody bonds IL-21 together with the free form of IL-21 or the IL-21 receptor bond IL-21 structure. And as we can see here, there are some residues show very substantial conformational changes upon the binding to IL-21 antibody or the IL-21 receptor. As we can see here, the gray one is the free form of IL-21, and this residue, lysine 73 and arginine 9, this particularly show the drastic conformational changes. And what I want to reassure you is that we are not doing some cherry picking here. Like we identify some residues, they have conformation change. So these two residues, lysine 73 and arginine 9, is actually very important for IL-21's binding with its receptor. So the previous crystal structure has shown that the mechanism how IL-21 interacts with its receptor is through forming these pocket structure to accommodate a methionine 70 residues from IL-21 receptor. So what happens here is that in the IL-21 receptor bound form, these two residues, they are kind of close to the methionine 70 residue in IL-21 receptor, so they can form this van der Waals force bond. But in the free form of IL-21, these two residues are just too far away from these key residues, methionine 70, and they cannot form any detectable contacts. What the antibody does is it forms a complex with IL-21 and pre-position these two key residues so that they can be close enough to this methylene 17, and they can readily bind to this residue. So the whole process just facilitates the binding between IL-21 and IL-21 receptor. So to further test the in vivo efficacy of this antibody, we develop a mouse model where we actually replace the mouse IL-21 and IL-21 receptor genes with the human counterparts, because the human IL-21 doesn't cross-reactive with the mouse receptor. And we test that in the mouse tumor model. So as we can see here, the IL-21 alone, it does not show any significant anti-tumor efficacy, which is aligned with some previous publication. But when IL-21 forms complex with the antibody, we can see a very unique anti-tumor efficacy. And we further repeat experiments in the earlier time points so that we can get some tumors in IL-21 antibody treatment groups and know what happens in the tumor microenvironments. As we can see here, the novel the IL-21 agonist antibody basically enhance the tumor-infiltrated T cell abundance for both activated CD8 T cells and CD4 T cells, but has less impact on Treg or NK cells. So we further profile the immune landscapes of tumor-infiltrated leukocytes and germ lymph node using single-cell RNA sequencing. And as we can see here, we further observe that agonistic IL-21 signaling can enhance the tumor-infiltrated CD8 T cell functionalities by inducing these cytotoxic cytokine genes. And it was confirmed by flow cytometry, as we can see here, the granzyme B producing CD8 T cells increase significantly with IL-21 antibody complex treatment. We also want to know what happens in germ lymph node, because they are essentially the reservoir for the tumor-infiltrated T cells. So we'd like to know what happens in the tumor germ lymph node. As we can see here, in the first place, the agonist IL-21 signaling can increase the abundance of germ lymph node CD8 T cells selectively. But we look into the heterogeneity of these germ lymph node CD8 T cells, we notice there is a specific cluster here, which is mainly contributed by these the purple subcluster 4, which is enriched in IL-21 antibody treatment group. We further analyze the signature of this cluster. We identify that they have very clear upregulation of IL-7 receptor, BCL2, which are the signatures of central memory-like phenotypes. And more importantly, to identify unique molecular signatures induced by agonistic IL-21 signaling in the germ lymph node CD8 T cells, we further analyze their differentially expressed genes compared to control groups. And this gene, Ly6A, really stands out. So Ly6A encodes a protein called stem cell antigen 1, SCAL1, which is considered as the marker for stem cell-like memory T cells. So indeed, in flow cytometry, combining with the central memory-like markers, we can identify a specific population which highly express SCAL1 and BCL2 in the IL-21 antibody treatment groups, indicating that these T cells in the germ lymph node might have improved stem-like memory phenotypes. So we further sought to compare the therapeutic efficacy of our agonist antibody with immune checkpoint blockade therapies. So as we can see here, both anti-PD-L1 therapy and our IL-21 antibody complex, they can both significantly suppress the growth of MC38 tumor. So this one, by this figure, you can see the significant statistical significant differences. And both the immune checkpoint blockade therapies and our agonist antibody can increase CD8 T cell infiltration to tumors, increase their cytotoxicity. But the important thing is that the IL-21 agonist antibody, they have some unique advantage, as they can induce some other cytotoxic cytokine expressing in the tumor-infiltrated CD8 T cells. So that indicates the potential for the synergistic function between the agonist antibody and the immune checkpoint blockade therapies. So in this project, we further develop this bispecific antibody, which we combine the anti-PD-1 and IL-21 agonist antibody into the same backbone. And we have validated that both arms' function has been maintained as we can see here, the bispecific antibody has the same function as our agonistic antibody to improve the human T cell activation. So further continuing this project, we are currently validating the therapeutic efficacy of this novel IL-21 agonism PD-1 blockade bispecific antibody in the lung cancer preclinical model. And this model was developed by Professor Nick Joshi in Yale University. They published this model first in 2021 in this Nature Biotechnology paper. So we are in collaboration with Professor Kunkun Khanna's lab in UK Mata Research Institute. We are now testing our bispecific antibodies efficacy in this tumor model. So the advantage of this model is that it can express some well-defined T cell antigen, which can mimic the lung cancer neoantigen, so that we can study the antigen-specific T cells phenotypes in this model. So to sum up, here we show that our agonist antibody can profoundly improve IL-21 function by optimizing its structure. So when this IL-21 antibody complex was treated to the mouse tumor models, they can amplify IL-21 signaling, such as STAT3, and promote stem-like memory, or at least central memory-like program in drained lymph node CD8 T cells, which can further enhance their supply of functional cytotoxic T cell infiltration into tumors, achieving better tumor suppression. At the end, I would like to acknowledge Q Cancer again for funding this project. I also would like to thank my team, my mentor, and all the great collaborators. Also, thanks amazing funding bodies from the UQ, from NHMRC Investigator Grant. Thank you very much.